I can imagine the cerebellum saying, well, that wall over there is pretty hard. Don't run your head into it. This cushion over here is nice and soft. You can sit on it. But it does it all, since the cerebellum's work is all unconscious, it's providing all the things you don't have to think about.
Hi and welcome everyone to our podcast Pioniers of Science, and welcome Lucas.
Hi Peter.
Well, Lucas, after 27 episodes, this is the first time I'm introducing you in English, and that's because I'm very pleased to welcome a special guest today who was so kind to join us from across the North Sea Channel. So from London, England, welcome to our podcast, Professor Richard Wingate.
Hi, really pleased to be here as well. Hi Lucas, hi Peter.
We're very pleased as well.
Yeah, indeed.
Richard, you are a developmental neurobiologist and professor at King's College London, and some years ago we also collaborated on a science project, which was fantastic to collaborate on. Last year you also published a book, The Story of the Brain in 10.5 Cells, a very intriguing title. I even saw it at the bookstore in Leuven. That's how I knew.
I'm going to have to stop there and say that's a complete surprise to me. I didn't know that.
Yeah, I saw I saw the book in Science and I saw your name and I thought, this is great. Okay, and so, yeah. Anyway, we appreciate that you can make some time to talk to us. And if I'm correct, you're going to introduce us to some intriguing aspects of the brain. But maybe to start, can you tell a bit more about yourself? What triggered your interest in science, in neurobiology, and how did that lead you to become a research scientist?
Yeah, no problem. I was originally a biologist at university, and I went into biology because it was somewhere where I could keep my drawing going, because I was always very interested in art, and just at school we did a lot of sketching of plants and animals, drawings and diagrams, and I think if I look back, that's really what sort of pushed me into biology. So I did biology at Manchester University, and I wasn't a very good student. I wasn't a bad student, but I wasn't particularly gripped. I did what I needed to do, but then maybe in my third year there was a lecture on chaos theory and population size, and how when something called the carrying capacity value reached 1.7, I think, the population sort of equilibrium became chaotic, and that just seemed so weird and so extraordinary that it just gripped my imagination. I just fell into studying harder and went to do a PhD from Manchester University at Oxford University, where I fell into, by accident, a very, very vibrant academic environment full of really good scientists, really excited by neurophysiology. So I wasn't really doing neurophysiology, I was looking at brain cell structure, and that again allowed me to draw quite a lot, and also look at chaos, because I used fractal dimensions to characterize or to measure how branched brain cells were. So they've got lovely branching structures, and how do you put a number on that so you can tell if things are changing or not, or how things change when the brain grows.
A bit of a combination of mathematics, art, and biology, then.
Yeah. So all three, and then I followed the normal path, PhD, going to do postdoctoral research. Again, intellectually exciting place, and that's where I've been ever since, really, for about 25 years.
The drawing already makes me think of Ramon y Cajal, this neuroscientist who is considered the father of neuroscience, who also did some detailed drawings of the brain and the brain structure. So that reminds me of him. I think maybe we're going to talk about him as well, I don't know. Because, as I said, you're a neurobiologist, so let's talk about the brain and maybe about the history of the brain. I don't know where you want to start the story, what intrigues you in the brain, what
Well, I think this. You mentioned Santiago Ramon y Cajal, and that's a really good place to start.
Yeah.
Because one of the intriguing things about brain cells was it was very difficult to convince the scientific world that brain cells existed, and Ramon y Cajal used a staining technique, which was named after the inventor of this staining technique. It's a way of making brain cells visible, and it was invented by Camillo Golgi, who was an Italian scientist. Cajal was Spanish, and when Golgi put pieces of brain, photography had just been invented, and the developing chemicals that we used had become available, and he thought, well, what would happen if I put some brain into photographic developer? And maybe he was imagining that maybe you'd see an image of the world on the brain, I don't know. But what he saw instead were these tiny thread-like structures and cell bodies scattered through brain tissue. The beauty of it was that his technique only labeled or only showed 1 in 1,000 of these brain cells. So what was normally completely densely packed, you just see either fragments, or if you're lucky, you see a whole brain cell just beautifully positioned there. And Golgi thought that it was one big interconnected network. But when Cajal saw the same material a couple of years after its invention, he saw individual brain cells. And that battle between, are there brain cells or is it one big mesh, continued for 20 years, at least, after their first discovery. Golgi and Cajal became the first scientists, first people actually to share a Nobel Prize. And I can tell you a little bit more of the story if you want.
I didn't know about that, actually, that they were the first people to win a Nobel Prize together.
Yeah,
Amazing
1901,
work as
I
well,
think.
So.
Yeah.
Oh, yeah, okay. Cool.
Yeah.
So they had a huge row, basically, when the prize was awarded. The awardees always had a special dinner and then gave a talk. And Cajal went one night and gave a talk. He was very complimentary about Golgi. He said, he invented this stain. It transformed Cajal's world. It made him completely dedicated to this journey of discovering what the brain is made out of. And then Golgi stood up the next night and just said, this is all rubbish. There's no such thing as a brain cell, basically. And it became very famous, actually, this disagreement.
Even though the concept of a cell was already invented in the century before, and I mean, the drawings are really beautiful. If you look at them, he really drew the tiniest details, that there is a gap, for instance, between a synapse and the next neuron. He saw that there was a tiny little gap that we now know is for neurotransmitter release. So it's really wonderful how we captured each detail of the brain, and
but I guess we've seen it often enough that it takes a while for people to believe such things.
I mean, that's the interesting thing, because of course, with the microscopes of the day, a synapse is angstroms wide, not microns. It's tiny gap. Cajal couldn't have seen that gap. And he said there must be a gap, whereas Golgi just didn't lift his pen up at the right moment. He just continued drawing, drew them connected. And so it was a real question of belief more than what you could see down the microscope. Cajal believed in the idea of a cell. Golgi didn't. Golgi didn't believe in that. He believed in a big network mesh, a kind of reverberating network of fibers, a bit like a giant fungus. Really.
Okay. And so how long did it take then until scientists started to consider the brain as an ensemble of brain cells? When did that change?
So it had already, before the... even before Cajal, there was starting to be some arguments that there must be brain cells. So there was an embryologist called Wilhelm His who saw at the beginning of development there are very simple cells, and globules in the brain that look separate. So I thought, well, if these are separate at the beginning, they must be separate at the end. And there was a French scientist called Auguste Forel who studied ants, was an obsessive about ants, and he kind of liked the idea, I think, of collective intelligence, that there could be individuals making big collective intelligence. Interesting Norwegian scientist called Fridtjof Nansen, who also won a Nobel Prize, but he won the Nobel Peace Prize much later on, who studied animals without backbones, and he was convinced that they were cells. And so there was already hints that cells, the brain was made of cells, the brain and spinal cord, but the majority believed that it was a network, just because you couldn't see anything else but this network. And it probably took about 20 years, I would say, for the network hypothesis to fade completely, I would guess. And Golgi never gave up on his idea that it was a network.
And did they have any idea at that point yet what the cells actually did, how they functioned, how they sent information, for example?
That really is Cajal's genius. He came up with a law which he called dynamic polarization, which kind of stated that information came in one end of a brain cell and exited at the other end. So something called the dendrite receives information, lots of dendrites in some cells, and then a single output, a part of the brain cell called the axon. And so if you apply that to all the drawings that he did, suddenly you see a network of information flow through the brain. And it's not just random. It's almost like an arrow saying information goes this way. And with that, he made huge progress just by doing these very intricate drawings. One thing I'd say about the drawings as well was, if you've ever looked at a stain with the Golgi stain, it's usually fragments, and Cajal must have spent quite a lot of time piecing it all together with his imagination. So it's not quite the objective scientific approach that you might think, although it is incredibly accurate. And Cajal himself acknowledged the fact that really there's a hell of a lot of subjectivity in science, in all science.
Yeah, especially if you look at this microscope as well, which was, a primitive instrument if you compare it to nowadays confocal microscopes, all the special things we have. He did it with a tiny thing. If you look at his pictures, that's really incredible what he did.
Yeah, they're incredibly detailed to indeed not actually be able to see a full cell properly at once, probably most of the time.
I don't think he saw very few cells would he have been able to see in their entirety. It just wouldn't have happened. So, and also just one other thing, I guess, about Cajal, the extraordinary thing is just how comprehensive he was in describing the brain. He looked at brains of lots of higher in inverted commas, let's say lots of mammals, and drawing comparisons between different species. Incidentally, his younger brother, who isn't very well known, Pedro, did all the reptiles and amphibians.
Okay, I didn't know that.
Yeah, he was a bit of a tearaway as a youth. But he ended up working with his brother and became also a kind of respected scientist and physician. But never had much of the fame.
Guess he remained a bit in the shadows of his of Santiago. Okay, and so, if we look at the brain now, I think the estimated number of brain cells is about 86 billion. So that's really amazing. But, as I remember well, I think, Richard, you also studied the cerebellum, which is a part of the brain that's located in the back of the brain. And half of the brain cells can be found in that tiny structure, which actually occupies only 10% of the volume. So can you tell us a bit more about the cerebellum and why it's so interesting to you?
Yeah, it's a very good introduction. Thank you. The cerebellum. Just to sort of amplify that a little bit, For most mammals, it's about half the total number of brain cells are in the cerebellum, this small, relatively dense, packed structure, highly folded, just like your cerebral cortex and stuck underneath actually the cortex. But in humans, it rises to 80%. So it's 60 billion cells in the cerebellum and only 80%. 20 billion cells in the rest of it. So the thing we think of when we think of the brain, we're actually thinking of the cortex. That's really relatively small amounts of the total brain cell number compared to the cerebellum.
Yeah, well, I feel a lot of the focus does go to that cortex when you're looking at research or when you're learning about the brain. A lot of the focus is on that outer layer and not necessarily the small, walnut-shaped cerebellum.
And there's a reason for that as well. Is even though the cerebellum's relatively simple in terms of its structure, it's only got really two major kinds of cell inside it, and if you sliced the cerebellum and looked at it anywhere, it would look the same. You wouldn't be able to tell where you are in the cerebellum. It always looks the same. It's got a repeated structure. No one really knows what it's doing. And the cortex is much more kind of easier to understand. It's got a map in it, different regions that do different things, parts of the cortex that recognize faces, another part that has memory, hearing. Visual cortex is divided into different areas that process different aspects of vision. It's all very nicely laid out, and it can be mapped with functional MRI, so you can see where different parts of the brain light up during different activities. The cerebellum's not like that at all. It's really complex, and the map, such that it is, of the body there's at least two of them, maybe four, perhaps, broken up into different pieces, sort of scattered along the surface. And it's a really mysterious object. It's funny that it's so ignored in the early brain scans. And you still see this. You do an MRI scan and it will come down from the top of the cortex, down, and stop just at the cerebellum because it's not important.
And so you said that was a bit the ancient view. So how does this view on the cerebellum as a simple, unimportant region, how did we acquire the view that it has maybe a lot more functions? How did we go through that in history? When did that change?
So it's a structure. It's got an interesting story, I think. Historically, it was very difficult to look at. It's very inaccessible. You couldn't really do experiments with it. If you want to call them ancient anatomists, people like Galen, who I think was a physician who, in Roman times, the gladiators, as one of his jobs, thought it was something that regulated the flow of cerebral spinal fluid, the fluid inside the brain. It was like a valve. That's what it was for. But maybe it also had some motor control functions. And that idea of it controlling movement is something that was reinforced by experiments, really early, very kind of gruesome experiments, where parts of the cerebellum were sliced away very slowly in pigeons until basically the pigeon couldn't move anymore. That's sort of around about the 1600s, and then it was refined, that idea that the cerebellum was responsible for, not for making movement happen, but just for making it happen well. And that was, again, reinforced by damage to human cerebellum, where you get a very characteristic movement tremor. When you've got damage on, say, the right-hand side of your cerebellum, it's the right-hand side of your body that will have something very characteristic, a tremor that gets worse as you try and pick up an object, and you lose your balance, you lose your stability. And so the cerebellum became associated with the idea it's to do with balance, it's to do with coordination, and also learning how to make coordinated movements.
And you mentioned there damage to the cerebellum, but can a human also survive without their cerebellum? Can you completely get rid of it, or is that too? I mean, would that kill you?
Yes, can they get rid of it? I'm trying to think, would that happen? I'm just thinking, could it happen? The answer is probably not. When you have one of the most common childhood brain cancers is medulloblastoma, which is a cancer of the cerebellum, basically. And it's not surprising it's common given how many brain cells are generated within the cerebellum. It's where the majority of brain cancers are in children. It's very, very difficult to survive the medulloblastoma, and the surgery is kind of not always negative results, but it's really difficult
the
to
impact
operate on without.
is really high,
The mortality rate is extremely high. So I was just going to say what was more revealing were gunshot wounds, particularly in the First World War. A scientist called Holmes tracked the effects on movement following rifle injuries to the cerebellum from soldiers in the First World War, and that was part of the argument, was that this is involved movement. I'm just going to add one more thing, actually, which you can be born without a cerebellum, and in that case, it's quite difficult to spot that you don't have one, and that is really remarkable. So,
and why is that? You mean spot on imaging, or spot it in time, for instance, during pregnancy, or you only can detect this later when you already born, for instance, that a cerebellum is missing? Or
so it's a really quite famous case which has upset a lot of people in the cerebellum world, that you can live without a cerebellum. This is not good if you work on the cerebellum.
Doesn't make it sound very important, no. If you can't even notice,
But there's a Chinese lady who was feeling sort of unsteady, unwell, headaches, persistent headaches, went to hospital, had a MRI scan, and they found she had no cerebellum at all. And it does happen. So my explanation would be that all the functions of the cerebellum can be taken over by the cortex, probably at huge cost in terms of energy and effort, because, for example, this Chinese lady learned to walk late in life. In many ways, very normal. I'd actually really love to follow that case up. It's just the single report, but I'd love to find out what other things she was experiencing. The argument that the cerebellum is important is that damage before birth, if you damage your cerebellum during pregnancy, then it's almost inevitable that you'll suffer some kind of cognitive disorder later in life. So it's very strongly associated with autism and other cognitive conditions. So damage it after you're born, you get a movement problem. That's the thing that's most obvious. Maybe there's a problem with cognition as well. If you damage it before you're born, then you have this very high chance of having later cognitive issues in life, and that's what I study.
Okay, because just to add something to that, apart from damage to the cerebellum, we can also have certain drugs that can also influence cerebellar function. And I think the most common example is alcohol, which can also sort of impair or limit a good function of the cerebellum, and also result in these movements that are not coordinated, and that's why the police always ask you to do the test to move your finger through your nose to your nose with your eyes closed, because the cerebellum actually guides the movement of your whole arm and your finger to your nose, and so when you're drunk, this function is completely imbalanced or impaired, and so you cannot bring your finger to your nose. Is that correct? Or I mean, there are also other regions involved, but
It's really it's something called the GABA receptors. So GABA is an inhibitor, and the receptor for GABA, they're disabled by alcohol. So and it's absolutely the case that everything you see with alcohol intoxication, and if you have long-term alcohol use, you have the same long-term effects on the cerebellum. The intention tremor, and also you're unsteady, you can't walk straight. So
So when you're chronically drinking alcohol, after a while you really damage the cerebellum as well.
Yeah, absolutely.
Okay.
Absolutely. All those are classic cerebellar signs.
Yes.
But the cognitive and emotional aspects of what the cerebellum does, that's more difficult to spot. I think partly because the movement disorders were so obvious and apparent. You just say, well, look. That's
because I was going to say, you've talked a lot about indeed movement and muscle control, which is something humans have, animals have, but as you mentioned at the start, our cerebellum takes up a lot more of our cells than in an animal's brain. So I was wondering, is there an explanation for that? Why do humans seem to put even more importance in their cerebellum than animals do?
Well, it's probably, and I think this is this will become clearer and clearer, that it's the cerebellar hemispheres, the bits at the side, which are disproportionately large in humans. So that's the central part, which is called the vermis. That's pretty similar in most mammals, but the hemispheres either side are much larger in humans, and the output, which is go from those hemispheres, is mediated. It goes through a small nucleus, well, not a small nucleus in humans, but a very large nucleus called the dentate nucleus. It goes to the prefrontal cortex. So the prefrontal cortex is the part of your cortex which reports or is active in a way that's related to your personality and to higher executive, as it's called, functions, so the higher decision-making functions of your brain. And it's the cerebellum, which is massively expanded. It's that part of the cerebellum which is hugely expanded. And that connection, that controlling, well, I'm going to say controlling, and you can talk about that if you want to, aspect of the cerebellum, the fact that the cerebellum has this connection to the cortex via another structure called the thalamus. The fact that it can do that's very much a mammalian feature of anatomy. So in non-mammalian animals, the connection from, or the information from the cerebellum really doesn't reach the forebrain, as it's called. It doesn't get up there in the same way.
And so that means there's much more to it than movement. So there are probably other things involved. So, and I think that's also work from recent years, that the cerebellum doesn't only control movement, but also has some other functions. Can you explain that?
So it definitely has these cognitive, emotional, in fact, the cerebellum is probably impacting, depending on which way you think about it, every part of the cortex. And so every function that you can think of, from vision through to making decisions, is influenced by the cerebellum. So the cerebellum is sending information. And it does that because the cortex is sending information to the cerebellum. So it's a circular loop. And you can also see this really clearly in the human brain. The connections from the output of your cortex to the cerebellum are hugely enlarged in the human brain. It's a part of the brain called the pons, and it's this big chunk of fibers, like a superhighway, that connects all the output from the cortex.
So they're actually constantly communicating. The frontal cortex and the cerebellum, they constantly in dialogue.
constantly in dialogue. There's another part of the brain also involved in that dialogue, which is the basal ganglia, but there's constant dialogue. Everything that happens in the cortex is fed through the cerebellum and back to the cortex. So that's the question, what comes first, the cortex or the cerebellum? And no one knows. No one knows the answer to that one. I did once have this conversation with a very famous anatomist, and I said, well, obviously the cerebellum is influencing the cortex. You know, it's shaping the way the cortex works, because the cortex is obviously the decision maker. But this is someone who died a couple of years ago, Ray Guillory, who was a very famous neuroanatomist, said, no, because the cerebellum is driving the cortex. The cerebellum has an excitatory output, and it drives the thalamus, which is also excitatory. So whatever it's doing, and the thalamus then drives the cortex. Yeah. It's a chain of excitation. So how it's working, I've got there are some other things it could be doing, but it's very mysterious.
Okay. So maybe the cerebellum is making the decisions up there in our head.
The way I think of it is maybe not making the decisions, but maybe focusing attention in a way. It's sort of saying this bit's important.
Yeah, because we always think about the brain as we see the world, and the cortex is sort of the endpoint of interpretation in the brain, and then directs an output to what we feel, think, or how we act in that world. But actually, that might be a too classical view. It might be completely different, with the cerebellum taking over some kind of decisions or impacting decisions more or less. It looks like the brain is composed of a lot of centers that communicate with each other, and one is always trying to get the overhand. I don't know. I don't know if that's a good representation, but
the way I like to think of it at the moment is the cerebellum is your world. It's it's kind of interesting that the cerebellum used to be seen as it's a single structure. It's a single organ. It doesn't have maps in it. It just is there to. In fact, there's a really nice way of thinking about it. Your body in space. It's your body in space, and this is ancient 1900s theory. But I quite like that. That the cerebellum holds an image of the world around you. It receives information about everything, and it predicts. It says, well, that I can imagine the cerebellum saying, well, that wall over there is pretty hard. Don't run your head into it. This cushion over here is nice and soft. You can sit on it. But it does it all. Since the cerebellum's work is all unconscious, it's providing all the things you don't have to think about.
Yeah, it's a bit that classic image of the baseball bat, and when you're training, when somebody throws a ball at you, the cerebellum helps you to learn how to hit it, and so it predicts the movement the ball is going to make, where it's going to end up, so that you can strike your baseball bat against the ball.
Yes.
That's maybe the simple example, but maybe it's true for the rest of what we live.
It's quite a good example, and I think it really says, it can sharpen that prediction. It can get better at doing it as you learn more and more. But it's doing the whole world.
Yeah, it's doing more than the ball.
It's doing the ball, but it's also watching the arm of the pitcher. It's making so in the end you don't have to. If you play baseball, which I don't, but you don't have time to think about where the ball is. You probably don't even see the ball, but you have seen the pitcher's arm. So these things are going at 90, 120 kilometers an hour. So it's actually more than just. My view, this is my personal opinion, is not finessing the muscle movement, it's finessing the world and your response to it. So the muscle movements come after it, but it doesn't control muscles, it's your cortex that controls the muscles. But it does say, well, the world is like this, and there's this thing moving towards you, and it's going to do this. So if you want to hit it, you do this.
You're going to do this,
Intriguing.
Because is there any question still that you'd like to see answered, or maybe even answer yourself in your research about the cerebellum? Or are there too many to choose from?
There are a lot to choose from. I'm really interested in this idea of the origin of those connections, when that connectivity happened in evolution, and how it happened. I guess that's one. What else am I interested in? I mean, so many things. Very, very interested in the animals that have very limited sensation, not only blind but burrowing in an earthworm-like way. So. Yeah. Not just making holes in the ground, but actually sort of squeezing through the ground. So they don't really have any kind of distance receptors as such. They just have body contact to feel their way. There's a whole class of legless or limbless amphibians that do this, and their cerebellums are really small. Understand that a bit better. And of course, the main research is the relationship between cerebellum development and autism, and trying to understand whether it's the connections that don't form in quite the same way as if you're non-autistic or at a different end of that spectrum. If the severity of, or how autistic a mouse would be in this case, is that related to the pattern of connections? That's probably main research that I'm doing at the moment.
Because that too is often linked to cortex problems. I mean, in neuroanatomy, that some connections are not well established in the cortex, but so what you propose is that in the cerebellum too, there are some anatomical problems, and so also physiological problems.
So there's some experiments that have shown that if you specifically knock out one class of cell, specifically in the cerebellum, you can create a mouse that has autistic-like syndrome. And that, if we go back a little bit, someone who's born without a cerebellum, doesn't seem to matter very much. If a cerebellum is there, it becomes this intimate partner with the cortex, and so if it's damaged, you get a breakdown in some of the function. So those connections are established remarkably early. They are probably one of the earliest sets of connections to be formed in the brain. So the output of the cerebellum is there before the cortex really develops.
Wow.
And so it's almost like the one way of thinking, the cortex gets hooked on the cerebellum at an early stage, and when after it's hooked, anything that disrupts that relationship can lead to, if it's before birth, it leads to cognitive disorders. Autism probably is, it's a condition. It's on a scale. Whether it's a disorder or not is a very kind of, what would be the word? Not controversial, but it's definitely something worth debating. I think people who are autistic would say, it's not a disorder. This is just the way I am.
Yes. Yeah. And so maybe a last thing about the cerebellum. So I mentioned in the beginning that you published the book 10.5 Brain Cells. And maybe one of the cells that you mentioned are the Purkinje cells. So one of the most beautiful and super large cell types in the cerebellum. And we also studied that together, I remember, at Leuven. That was very nice to see. So anything there that fascinates you about this cell type? I mean, it's really beautiful.
I'm thinking back to your work, actually. Going back and thinking, this could be really interesting. We looked at the development of these cells using the same kind of techniques that you and Joka used back in the day in Leuven. And they're very interesting, the way they grow and migrate. So brain cells are born in one place, and then they have to migrate into their final position. And Purkinje cells undergo this really dramatic change. They move themselves, they sort of crawl up from where they're born, upwards and upwards and upwards. And when they finally find their place, they lose all their intricate dendrites, the branches that make them so characteristic, and they grow a whole new tree. So They undergo this really from kind of larval Purkinje cell to fully fully
it
beautiful.
really looks like a
Purkinje
tree.
cell.
Think,
And it looks like a tree.
anyone who's listening can maybe type it in on Google, Purkinje cell, and you will see like you have the small nucleus, so this the cell core, let's say, and on top of it is a giant tree. I mean, it's really wonderful to see. It's really an intriguing cell type. I think it's one of the largest cells in the human brain. I don't know.
It's
Or the
certainly
most
got one of the
complex.
largest. It's got one of the largest dendritic trees.
Yeah.
There are some absolutely monstrous cells though.
Yeah,
I mean,
in terms
but-
of distance, true. Yeah.
yeah.
But the tree is really amazing.
And again, what it does, it's really, really just it's. I would say, I didn't start off in the cerebellum, and I came into it relatively late, and I'm always fascinated by going to a cerebellum conference because there are only 3 or 4 kinds of cells to look at, and they're studied in such intricate detail. But the overall picture, has somehow been lost in the. To use the expression, you can't see the wood for the trees. It's the overall picture of what the cerebellum does is somehow kind of almost forgotten in the absolute detail of trying to work out how the connections are working, what the little tiny circuits are doing, how memory traces are formed. And these are all very interesting things. Memory as in how a activity in a brain cell can shape its future activity, so it's going to change its activity depending on what signals it's received. But the overall picture is rarely discussed. It's a really interesting place to be, and I think, as you said, Lucas, everyone thinks about the cortex. So it's a very maybe it's a kind of enthusiasts area, but I have hope that the cerebellum will become more well known, I guess.
Well, I guess you being here has already helped that a bit, so thank you very much for joining us on the podcast as well to talk about this often forgotten little brain. So with that, I think we can maybe wrap up there. Thank you very much for being here.
Thank you very much. I've really enjoyed the conversation.
Yeah, thank you.
Yeah, me too. And thank you again, Peter, of course, for being here as well.
Yes.
And then we'll see everyone again in 3 weeks' time. So see you then.
See you then. Bye-bye.
Bye.